Welcome to Quietly Made. I am so glad you have found your way here tonight. This is a space for the curious and the weary, a place where we explore the vast, invisible infrastructures of our world, while giving your mind the permission it needs to simply rest. Before we begin, I invite you to join our community by subscribing and leaving a comment. I often wonder where these stories find you tonight. If you feel inclined, please leave a comment sharing what part of the world you are in and the hour you are settling into sleep. It is a small way for us to feel the quiet connection of this community, scattered across different time zones, but sharing this same moment of rest. If you fall asleep, that is exactly what should happen. You are safe here. You can always come back tomorrow and pick up exactly where you left off. Adjust your pillow. Take a deep, slow breath in and let it out. Let your mind drift as we begin. Tonight, we turn our attention to the quiet assembly of something smaller than a grain of pollen and older than any language ever spoken. We turn to the code written inside every living thing and to the long, patient story of how a curious species began to read it and then, very carefully, to touch it. Long before anyone understood what a cell contained, people were already editors of life in the only way available to them. Think of a field at the edge of a river valley, the soil dark and heavy after a season of rain. A farmer walks the rows at dusk, running a thumb along the husks of grain, feeling for the ones that are fuller, heavier, less likely to shatter in a strong wind. There is no microscope in this scene. There is no laboratory. There is only touch and memory and the accumulated attention of many hands across many years. Seed by seed, the farmer is making a decision about the future without knowing that decision has a name. This is where the story begins, not with glass and steel, but with soil and patience. The earliest editing of living things was slow beyond imagining, measured not in hours, but in generations. A grain that yielded a little more, a fruit that ripened a little sweeter, an animal that carried a little more meat on its frame. These small differences were noticed, and noticing became selection. And selection, repeated across 1,000 seasons, became something that looked almost like design, though no one alive at the time could have called it that. Consider the texture of an ancient granary, the way dried kernels shift and settle when poured, the faint dust that rises and catches the low light of an oil lamp. Someone standing there, centuries ago, choosing which sacks to set aside for next year's planting, was already making a wager about heredity. They could not see the thread that carried the instructions from one generation to the next. They could only see its shadow, cast across the fields and the pens and the orchards, in the shape of a slightly better harvest, a slightly hardier herd. The need to understand this hidden thread did not arrive all at once. It gathered slowly, the way frost gathers on a window overnight, crystal by crystal, until the whole pane is covered and the pattern can finally be seen. Disease played its part. A field wiped out by blight in a single season taught a harder lesson than any success ever could. a herd thinned by a sickness that seemed to choose its victims by some invisible rule, taught people to ask why. The answer, they suspected, was carried inside the animal itself, passed down like an heirloom too small to see and too important to ignore. There is a particular kind of quiet that settles over old agricultural records, the ledgers kept by hand, listing which parent produced which offspring, which lines were kept, and which were let go. Each entry is a small act of faith in something unseen, a belief that traits travel, that a bloodline is a kind of sentence written in a language not yet decoded. Meanwhile, in kitchens and breweries and orchards, another form of unconscious editing was taking place. A baker who saved a piece of dough to start the next batch, allowing the same population of tiny organisms to multiply and ferment and rise again, was cultivating life without knowing it. The bubbles forming in the dough, the faint sour smell rising from the crock, the way the texture changed from stiff to elastic overnight, these were the visible signs of an invisible process, populations of living things being shaped, generation after generation, by the conditions humans chose to provide. It would be easy to imagine this early period as primitive, a mere prelude to the real story. But that would miss something essential. The instincts formed in these centuries, the idea that traits can be selected, that offspring resemble parents in patterned ways, that careful choice repeated over time can reshape a living population, became the foundation on which every later discovery would rest. No laboratory, however advanced, invented the concept of heredity. It was inherited, so to speak, from those quiet fields and granaries, from farmers and herders and bakers who never saw a cell, yet somehow, through patience alone, learned to work with the grain of life itself. As the light fades further into evening, imagine the weight of an old wooden yoke resting on the shoulders of an ox chosen generations back for its calm temperament, a trait selected for so long it had become almost as reliable as its size or color. Imagine the smell of straw and warm animal breath in a barn at the close of day. In that ordinary scene lies an extraordinary process, waiting for someone someday to ask what it was actually made of. The need for a deeper understanding grew heavier with time, the way a held breath grows heavier the longer it is kept. It was not enough anymore to simply notice that traits repeated. People wanted to know why. They wanted a mechanism, a reason, something they could hold in their minds the way they held a seed in their palm. Picture a stone terrace overlooking cultivated hillsides. Someone standing there in the cool of early morning, tracing with their eyes the differences between one row of vines and the next. Some vines had thrived through a dry season that had withered others planted only a few paces away. The soil was the same. The rainfall was the same. The only difference lay somewhere inside the vines themselves, an inheritance too fine to see, yet decisive enough to determine survival. Moments like this, repeated across countless fields and countless generations, planted a different kind of seed, the seed of a question that would not let go. Illness sharpened this question in ways that comfort never could. A community touched by a condition that seemed to run in families, appearing in one line and skipping another, presented a puzzle that could not be explained by chance alone. Physicians of earlier eras, working without any of the tools we take for granted now, kept careful notebooks recording which afflictions clustered in which households, which conditions seemed to travel along bloodlines like water finding the lowest point in a landscape. These notebooks, dense with handwriting and crossouts, are themselves a kind of monument to need. They represent thousands of hours spent trying to see a pattern that remained frustratingly just out of reach. There is a particular texture to old paper that has been consulted many times. The softness at the corners, the faint darkening where fingers have turned the pages again and again. Somewhere in an archive, pages like these still exist, filled with careful tables, comparing the traits of parents and offspring across dozens of families, an attempt to reduce the mystery of heredity into something structured enough to study. The ink has faded in places, but the intention behind it has not. Every entry asks the same unspoken question: what is being passed down, and how? Meanwhile, in workshops where lenses were carefully ground and polished, another kind of need was taking shape. As instruments for seeing small things slowly improved, curious observers began peering into worlds that had never been visible before. A drop of pond water, magnified and held still beneath careful light, revealed shapes moving with purpose, structures no one had imagined existed. The realization that living things contained entire hidden universes at scales too small for the naked eye completely reordered what people believed was possible to know. If there were worlds this small already visible, what other worlds might still be waiting, one lens improvement away? Consider the discipline required to grind a piece of glass by hand for hours, checking the curve against the light again and again, all in service of seeing something no one had seen before. This same discipline, patient, repetitive, devoted to a goal that might take a lifetime to reach, would define the centuries that followed. The need to understand heredity was never going to be satisfied by a single flash of insight. It would require thousands of small refinements, the way a lens is ground, not in one motion, but in 10,000 tiny adjustments. By the time this need had fully taken hold, it had spread far beyond farms and breweries and physicians' notebooks. It had become a question asked in gardens, tended for study rather than food, in the careful crossbreeding of ornamental plants chosen purely to observe what patterns would emerge. It had become a question whispered in the quiet hours after a difficult birth, in the careful watching of a newborn's features for traces of one parent or the other. It had become, in short, one of the great unanswered questions sitting beneath the surface of everyday life, waiting patiently, the way a seed waits beneath frozen ground for a season warm enough to let it grow. As this chapter closes, imagine the hush of a garden at the end of a long afternoon of careful observation, rows of plants marked with small wooden tags, each one recording a cross made deliberately, a question posed directly to nature itself. The light is beginning to fade. the observer closes a notebook filled with careful counts and measurements, unaware that these humble records would one day become the first true glimpse into the hidden architecture of inheritance. The need had been gathering for centuries. Soon, it would begin to find its shape. Understanding does not always arrive with noise or spectacle. Sometimes it arrives quietly, in the corner of an ordinary garden, tended by someone with more patience than ambition. Picture rows of pea plants growing along wooden trellises, their tendrils curling delicately around each support. Their flowers are soft, unassuming purple or white. This modest setting, unremarkable to any passerby, would become the place where the first real structure was given to an idea that had, until then, remained frustratingly formless. The appeal of the pea plant lay in its simplicity. Its traits were easy to see and easy to count. A pod was either smooth or wrinkled. A flower was either one color or another. A stem grew either tall or short. There was no ambiguity, no blending of qualities into a confusing middle ground. This clarity made it possible, for the first time, to actually count outcomes across many generations, to move beyond vague impressions and towards something that resembled a real pattern, something almost mathematical in its precision. Imagine the careful, repetitive labor this required. Each morning, walking the rows with a small brush or a pair of tweezers, transferring pollen by hand from one chosen flower to another, taking great care to prevent any stray pollen from interfering with the cross. This was not glamorous work. It demanded steady hands and steadier patience, the willingness to repeat the same delicate motion hundreds of times across an entire season, and then to wait, through weeks of quiet growth, for the results to finally reveal themselves in the shape of the next generation's pods and flowers. When the results did appear, they were startling in their consistency. Certain traits seemed to vanish in one generation only to reappear, unchanged, in the next. It was as though some traits were quietly waiting their turn, hidden but not erased, present but not expressed. This observation alone was enough to overturn a great deal of earlier thinking, much of which had assumed that traits from two parents simply blended together, the way two colors of paint might mix into a single new shade. What these garden rows revealed instead was something closer to discrete units, individual instructions that remained intact even when hidden from view, passed along whole rather than diluted. Consider the quiet satisfaction of counting, session after session, the ratios in which certain traits appeared across hundreds of offspring. 3 of one kind to every one of another, again and again, across different traits and different crosses, the same simple ratio emerging with a consistency that felt almost too orderly to be coincidence. This kind of numerical regularity, found not in the stars or in falling objects, but in the humble structure of a garden pea, suggested that heredity followed rules every bit as precise as the rules governing the physical world. It was not chaos. It was not mere chance. Somewhere beneath the surface, something orderly was at work. The notebooks kept during these years are a study in devotion. Page after page of careful tallies, columns of numbers representing individual plants, individual pods, individual seeds, each one a small data point in a much larger pattern. There is something moving in the sheer volume of this record keeping, the evidence of thousands of hours spent counting small, unremarkable things in the belief that within that tedium lay a truth worth uncovering. It is a reminder that some of the most important discoveries in the story of life were not made through sudden flashes of genius, but through the slow, unglamorous accumulation of careful observation. Outside the walls of this particular garden, the wider world of the time had little interest in what was being discovered there. The significance of these findings would not be recognized for a very long while, tucked away in journals that few people read, waiting patiently, much like the traits themselves, to be rediscovered by a later generation, better equipped to appreciate what they meant. There is something quietly poignant in this delay, an idea correct and complete, sitting in near silence for years, waiting for the rest of the world to catch up. As evening settles over that same modest garden, imagine the trellises casting long shadows across neat, orderly rows, the pea pods swaying gently in whatever breeze moves through at dusk. Somewhere among those unassuming plants, an idea had taken root that would eventually reshape how an entire species understood its own inheritance. The soil was ordinary, the plants were ordinary, but the pattern hidden within them was anything but. Ideas, once given shape, do not always travel quickly. The careful counts and ratios discovered in that quiet garden remained largely unnoticed by the wider world for a long stretch of years, resting in scientific journals like a seed resting beneath frozen soil, present but dormant, waiting for conditions to change. Elsewhere, other observers were approaching the same mystery from entirely different angles, unaware that a solution already existed in a paper few of them had read. Some studied the microscopic structures inside cells, watching through improved lenses as strange, thread-like shapes appeared and disappeared during cell division, condensing and separating in ways that seemed deliberate, almost choreographed. There was something hypnotic about watching these structures align and pull apart, a silent dance performed inside every dividing cell, following rules that no one yet understood, but that were clearly, unmistakably, rules. Picture a darkened room, lit only by a single lamp, its light directed down through a series of ground lenses onto a thin, stained slice of tissue pressed between two pieces of glass. An observer leans close, adjusting the focus by fractions of a turn, watching thread-like shapes emerge from what had moments before looked like uniform, formless material. The concentration required for this kind of observation is total. A held breath, a steady hand, the smallest movement of the focusing wheel revealing structures that had been present all along, simply too faint, too fine, too small to notice without exactly the right adjustment of light and glass. As different threads of investigation slowly converged, a picture began to form of a hidden architecture inside every cell, structures that seem to carry something essential from one generation of cells to the next, doubling before division, splitting evenly, always returning to a stable and consistent number within a given species. This regularity was itself a clue. Whatever these structures were, they behaved with a discipline that living things had shown before only in the counted ratios of pea plants and garden crosses. The observers of cells and the observers of traits were, without quite realizing it, circling the very same truth from two completely different directions. Consider the slow accumulation of small technical improvements that made this convergence possible. Better stains that made faint structures suddenly visible. Better lenses that resolved detail previously lost in a blur. Better methods for slicing tissue thin enough to let light pass through cleanly. None of these improvements were dramatic on their own. Each was a modest refinement, a slightly better dye, a slightly sharper lens, a slightly thinner slice. Yet stacked one atop another across years of patient technical work, these small refinements opened a window that had simply not existed before. And through that window, an entirely new layer of biological reality came slowly into focus. There is a particular kind of excitement that belongs to this era, quieter than the excitement of sudden discovery, but perhaps more durable. It is the excitement of pieces slowly fitting together, of separate lines of evidence beginning to point toward the same conclusion from different directions. A researcher studying inheritance patterns in one country might have no knowledge of a researcher studying cellular structures in another. Yet both were, in their own way, describing the same hidden mechanism, one from the outside, through counted traits and predictable ratios, and one from the inside, through stained slides and careful microscopy. When the earlier garden studies were finally rediscovered, years after they were first published, they landed on a scientific community far better prepared to understand their significance. The structures being watched inside dividing cells suddenly had a purpose that made sense. The ratios counted in generations of pea plants suddenly had a physical location, a place inside the cell where those units of inheritance might actually reside. Two separate strands of investigation, one mathematical and one visual, wove together into something stronger than either had been alone. As this chapter of understanding drew to its close, imagine the quiet triumph of a scientist finally holding both threads of evidence at once, the counted ratios in one hand, so to speak, and the observed cellular structures in the other, recognizing for the first time how completely they belong together. It was not a loud moment. There was no fanfare. It was simply the quiet click of a puzzle piece finally settling into its rightful place, the culmination of decades of patient counting, patient observing, and patient waiting. Once the outline of the mechanism became clear, a different kind of work began, slower and more painstaking than anything that had come before. It was one thing to know that traits were carried by structures inside the cell, passed faithfully from one generation to the next. It was quite another to understand what those structures were actually made of and how they managed to carry so much information in a form small enough to fit inside a space too tiny to see without powerful magnification. Picture a laboratory bench cluttered with glassware, flasks connected by narrow tubes, the faint hiss of a burner keeping some mixture at a steady, careful temperature. Researchers of this era spent long hours extracting material from cells, refining it through repeated washes and separations, trying to isolate the substance responsible for carrying hereditary information from the many other substances also present inside every living cell. This was slow, exacting labor, the kind that could consume entire careers without producing a definitive answer, and often did. There is a particular tension that belongs to this period of accumulation, the sense of being close to something enormous without quite being able to see its full shape. Different laboratories, working with different techniques, slowly built up separate pieces of evidence, each one suggestive but incomplete on its own. Some experiments pointed toward one type of molecule as the likely carrier of heredity. Other experiments seemed to point elsewhere. The evidence accumulated unevenly, the way sediment builds up unevenly at the bottom of a slow-moving river, thicker in some places, thinner in others, taking shape gradually, rather than all at once. Consider the discipline required to run the same experiment dozens of times, adjusting one small variable after another, searching for the exact condition under which a clear result might finally emerge. A researcher might spend months preparing a single careful demonstration, only to find the results ambiguous, requiring the entire process to begin again with some small refinement. This kind of repetition can feel, from the outside, almost indistinguishable from failure. Yet, within the world of careful science, it is simply the ordinary texture of accumulation, each repetition narrowing the range of possible explanations, even when no dramatic breakthrough seems to have occurred. Picture the quiet satisfaction of a single afternoon in which one particular experiment, involving the deliberate transfer of material between two different strains of the same organism, produced a result that could not easily be explained by anything other than a very specific carrier of hereditary instruction. The glassware involved was unremarkable. The technique was painstaking rather than elegant. And yet, the implications rippled outward through the scientific community. Another small but decisive piece settling into place within a much larger, still incomplete puzzle. As more evidence accumulated from more directions, chemists began to characterize the physical properties of the substance now suspected of carrying heredity. They measured its behavior under different conditions, its consistency, its response to heat, and to various chemical treatments. Slowly, a physical portrait began to emerge. Not yet a complete structure, but enough of an outline to know that whatever this material was, it was remarkably stable, remarkably consistent from one organism to the next, and present in quantities that scaled sensibly with the complexity of the organism it came from. There is something almost meditative about this period, an entire scientific community working in parallel, each laboratory contributing a small piece of evidence without any single person holding the full picture. Letters pass between researchers across long distances, describing results, questioning interpretations, proposing new experiments to resolve remaining doubts. The slow rhythm of this correspondence, penned by hand and carried by mail across weeks rather than seconds, gave each finding time to be considered before the next was added to the growing body of evidence. As this stretch of patient accumulation draws to its close, imagine a shelf lined with carefully labeled glass vials, each one containing a small sample extracted through months of careful work, each one representing a fragment of evidence pointing toward the same hidden answer. The full structure of what these vials contained remained for now just out of reach, but the outline was becoming unmistakable, the shape of something vast and essential, slowly emerging from years of quiet, careful accumulation, the way a shoreline slowly emerges from fog as morning light continues to strengthen. The shelf of labeled vials from the previous stretch of work did not stay still for long. Once chemists had a strong sense of what carried heredity, a new question rose to take its place, quieter but far more demanding. If this material held the instructions for an entire living thing, how was it built? Not just what it was made of, but how those parts were arranged, and why that arrangement could hold so much information in a space too small to see without the strongest lenses available. Picture a room lined with dark curtains, kept intentionally dim, because the equipment used to study this question relied on the faint scattering of light passed through crystallized samples. A researcher works for hours in near darkness, adjusting a beam by fractions of a degree, capturing the resulting pattern on film that must then be developed with equal care. The pattern itself looks like nothing at first glance, a scatter of dark points arranged in a shape that means nothing to an untrained eye. And yet, within that scatter, if read correctly, is a kind of geometric fingerprint, a clue to the underlying architecture of the molecule that produced it. Consider the frustration and the thrill bound together in this kind of work. Weeks might pass between one usable image and the next, the crystal sample degrading, the exposure imperfect, the pattern too faint to interpret with confidence, and then, unpredictably, a single image would emerge sharp enough to study properly, an X shape traced across the film, symmetrical and unmistakable, suggesting for the first time a structure built from two intertwined strands rather than one. That single photographic plate would eventually become one of the most consequential images in the entire story, though the person who produced it could not have known that in the moment it was taken. Elsewhere, model builders worked with an entirely different kind of material, twisting wire and cutting metal plates into shapes meant to represent atoms and the bonds between them, physically assembling possible structures on a tabletop, the way a child might assemble a puzzle. Every proposed arrangement had to satisfy a long list of chemical constraints, the correct distances between atoms, the correct angles of bonding, the correct fit between one component and the one beside it. Building the wrong structure was easy. building one that satisfied every constraint at once required patience, precision, and no small amount of trial and error. There is something almost tactile about this period of discovery, an unusual blend of physical craftsmanship and abstract reasoning. Imagine the sound of small metal pieces clicking into place on a laboratory bench, adjusted and readjusted. A base tipped slightly one way, then another, checking whether it still fit against its neighbor. Days were spent this way, assembling and disassembling, testing one configuration after another against the evidence gathered from those faint, scattered patterns of light. It was a puzzle with physical stakes, solved not purely on paper, but with actual hands moving actual pieces of metal and wire. When a configuration finally emerged that satisfied every piece of evidence at once, two long strands winding around a shared axis, connected by a repeating sequence of paired components running between them like the rungs of a twisted ladder, something shifted in how heredity could be imagined. It was no longer an abstract concept, a mysterious carrier of traits passed along in some unspecified form. It had a shape now, elegant and specific, a shape that immediately suggested how the material might be copied, one strand serving as a guide for the reconstruction of its partner over and over, generation after generation. Consider the particular quiet of the moment this structure was first sketched, in pencil, on ordinary paper, the two winding lines and the connecting rungs drawn out clean and simple after so many false starts. A structure this elegant, once seen, felt almost inevitable, as though it could not have been any other way, even though reaching it had required years of faint photographic plates, painstaking model building, and the quiet correspondence of researchers working across different institutions toward the very same unseen target. As this chapter of structural discovery settles into its final hush, imagine the stillness of a laboratory late at night, the models left standing on the bench exactly as arranged at the moment the last piece clicked into place, catching the low light of a single desk lamp. The twisted shape sitting there in metal and wire was no longer just a guess. It was, at last, a blueprint, waiting for the next generation of careful hands to learn how to read it, and eventually, how to begin working with it directly. A blueprint is not the same as a set of tools. Knowing the shape of something is one kind of understanding. Learning to reach inside that shape, to cut it precisely and join it deliberately, is another kind entirely, and it would take a different sort of patience to arrive there. Picture a laboratory bench, crowded with small glass tubes held upright in metal racks, each one containing a clear liquid kept carefully chilled on a bed of ice. Inside those tubes floated molecules extracted from ordinary bacteria, unremarkable organisms found in soil and water everywhere, yet carrying within themselves a peculiar and useful defense. These organisms had developed over countless generations a way of recognizing and cutting foreign genetic material that intruded into their cells, a kind of internal immune response, operating at a molecular scale. Researchers slowly realized this natural defense could be borrowed, removed from its original context, and put to work as a precise, reusable cutting tool. Consider the specificity required for such a tool to be useful. It could not simply cut anywhere at random. It needed to recognize a particular short sequence, a particular pattern within the long, twisted strands, and cut only there, consistently, every single time. Discovering which of these natural cutting tools recognized which sequences required years of careful, repetitive testing, exposing samples to different extracted enzymes and observing exactly where the resulting fragments broke apart. Slowly, a growing library began to take shape, a catalog of tools, each one keyed to a different short sequence, each one capable of making a clean, predictable incision at exactly the site required. Imagine the particular satisfaction of watching this kind of clean cut confirmed for the first time. The fragmented pieces separated by size using a shallow tray of gel through which a gentle electrical current was passed, causing the fragments to migrate at speeds determined by their length. Under a soft ultraviolet glow, the fragments appeared as faint, glowing bands arranged neatly by size, a visible confirmation that the cutting tool had done exactly what it was expected to do. There is something deeply satisfying about a prediction confirmed this way, an invisible molecular event translated into a simple, visible pattern of glowing bands in a darkened room. Cutting, however, was only half the task. A tool this precise would be far less useful without an equally precise way to rejoin fragments afterward, to take a piece cut from one source and attach it deliberately to a piece cut from another, creating a new combination that had never existed before in nature. Here again, researchers turned to existing biological machinery, borrowing another kind of molecule whose ordinary function was to repair breaks in genetic material, sealing gaps back together as part of the normal, continuous business of cellular repair. Repurposed carefully, this same repair machinery could be used to fuse two fragments from entirely different sources into a single, continuous strand. Picture the delicate choreography required to combine these two tools into a single deliberate act, cutting a fragment from one source with careful precision, cutting a receiving fragment from another source at a matching site, and then allowing the sealing tool to join them cleanly at their shared edges. The chemistry involved a careful balance of temperature and timing, brief incubations measured by the second. The mixture kept just warm enough for the necessary reactions to proceed without allowing anything to degrade. Small errors at any stage could ruin hours of careful preparation. There is a particular kind of quiet triumph in the earliest successful combinations of this kind. Fragments from entirely unrelated sources joined into a single continuous strand for the first time, a combination that existed nowhere in the natural world until a careful hand assembled it deliberately on a laboratory bench. It represented something genuinely new, not the discovery of an existing pattern, as the earlier chapters of this story had been, but the deliberate construction of a pattern that had never existed before. The instructions of life, once only readable, had become, in a very real sense, writable as well. As this chapter draws toward its close, Imagine the low hum of a small centrifuge spinning down a final sample, the faint clink of glass tubes settling into their rack, the quiet satisfaction of a long day spent perfecting a technique invisible to the eye, but unmistakably real in its consequences. The tools for cutting and joining had arrived. What remained was to learn precisely how to use them, and with a growing sense of the responsibility that came with them. With cutting and joining now possible, a new set of practical questions rose to take their place. A fragment could be cut and combined with another, but a combination sitting inside a glass tube accomplished little on its own. For this new instruction to matter, it needed a way to be carried into a living cell, copied faithfully as that cell divided, and ultimately expressed, translated into the working molecules it described. Picture a small flask of bacterial culture, kept gently agitated on a shaking platform. The liquid inside cloudy with countless individual cells multiplying steadily in warm, nutrient-rich broth. Researchers discovered that under certain carefully controlled conditions, a brief shock of temperature or a specific chemical treatment could make the outer membrane of these cells temporarily porous, just permeable enough to allow a small circular loop of genetic material to slip inside. This process demanded exact timing. Too little exposure and nothing would pass through. Too much and the cells themselves would not survive the treatment. Somewhere in that narrow window sat a small percentage of cells that took up the new material intact and lived to divide again, carrying the newly introduced instructions forward into every subsequent generation. Consider the almost absurd scale of the numbers involved in identifying which cells had actually succeeded. Out of countless millions of individual cells treated in a single batch, only a small fraction would take up the desired combination successfully. Researchers developed clever methods for finding this needle within such an enormous haystack, incorporating markers into the introduced material that made successful cells resistant to certain compounds, while leaving untreated cells vulnerable. Spread across a shallow dish coated with the right nutrient gel and the right selective compound, only the successful cells would survive and multiply, forming small, visible colonies scattered across an otherwise empty surface. Each colony a confirmed success story, grown large enough to see with the naked eye. Picture the quiet moment of checking such a dish after an overnight incubation, holding it up to the light, counting small round colonies scattered unevenly across the surface like stars across a clear night sky. Each visible colony represented not just a single successful cell, but the entire genetic combination carried faithfully through countless rounds of division, multiplying by simple doubling until it had become millions of identical copies, all descended from that one original successful event. There is something genuinely moving about this kind of exponential growth, an event that began as a single, uncertain molecular encounter blooming within a single day into an entire visible colony carrying the very same instruction in every one of its countless cells. As this technique matured, researchers began using it not merely to demonstrate that combination was possible, but to accomplish something with genuine practical value, introducing instructions for producing specific, useful compounds that these microscopic cells could then manufacture at scale, functioning as tireless, microscopic production lines running continuously in warm, gently shaking flasks. What had once required extraction from far scarcer and more difficult natural sources could now, in principle, be produced reliably, in quantity, by careful cultivation of cells carrying the appropriate instructions. There is a particular texture to laboratories from this period. Glassware everywhere, shaking incubators, humming steadily in the background, shallow dishes stacked in refrigerators awaiting the next morning's count. Careful notebooks recorded each attempt, most of them modest failures or partial successes, a handful of them genuine breakthroughs. The work was slow, methodical, and largely invisible to anyone outside the laboratory walls, yet it was steadily assembling the practical foundation for everything that would eventually follow. As this chapter of mechanical mastery comes to its quiet close, imagine a researcher standing at a bench late in the evening, holding a small dish up toward the light one final time before placing it carefully in the incubator overnight, already anticipating the count that morning would bring. The instructions of life could now be cut, joined, carried into a living cell, and copied faithfully across countless generations. What remained was to make this careful, delicate process reliable enough and simple enough for it to spread far beyond the walls of any single laboratory. A technique that works once in a single careful laboratory is not yet a technique that can be trusted broadly. For these methods to matter beyond the walls where they were first developed, they needed to become something else entirely, dependable, teachable, repeatable by careful hands anywhere in the world. Not just by the small number of researchers who had spent years perfecting every small detail through trial and error. Picture a shared laboratory notebook, photocopied and mailed between institutions, containing painstaking descriptions of exact temperatures, exact timings, exact concentrations of every reagent involved. Early attempts to reproduce a promising result in a different laboratory often failed for reasons that seemed, at first, mysterious. A slightly different batch of chemical reagent. A water bath calibrated a few degrees differently than the original. A glass tube of a slightly different thickness that conducted heat at a different rate. Or a bench positioned nearer a drafty window than the sender's own. Each of these small, easily overlooked variables could mean the difference between success and failure, and identifying them required careful, systematic troubleshooting, often conducted entirely through careful written correspondence between distant institutions, letters crossing back and forth for weeks before the discrepancy was finally traced to its source. Consider the significant labor involved simply in standardizing the tools themselves. The cutting enzymes borrowed from bacteria needed to be purified consistently, batch after batch, with reliable activity that did not vary unpredictably from one preparation to the next. Researchers spent considerable effort refining extraction and purification methods until these enzymes could be reliably produced, tested for consistent activity, and eventually shared or sold in a form stable enough to survive shipping and storage without losing their essential function. What had once been a delicate, custom preparation, made fresh in a single laboratory, slowly became something closer to a dependable, catalog-listed reagent. Picture the careful work of assembling reference materials, small circular carriers engineered specifically to hold introduced genetic instructions efficiently, tested and refined across many iterations until certain versions proved reliably superior, easier to introduce into cells, more stable once inside, less likely to be lost during cell division. These engineered carriers became something like standardized vessels, shared freely between laboratories, each one representing dozens of earlier attempts that had been quietly discarded in favor of a design that simply worked more consistently than its predecessors. There is a particular kind of generosity that belongs to this period of standardization. Laboratories mailing small samples of their carefully purified tools to distant colleagues, sharing exact protocols without asking for anything in return beyond an acknowledgement in a future publication. This informal network of exchange, built on trust and shared curiosity rather than any formal system, allowed methods first developed in a single location to spread gradually outward, refined slightly by each new laboratory that adopted them, each contribution folding back into a growing, shared body of practical knowledge. Imagine a young researcher, newly arrived at a laboratory, being handed a thick binder of accumulated protocols, page after page of careful instructions refined by years of collective trial and error. Reading through such a binder is like reading a kind of communal memory. Every small note about which step tends to go wrong, which reagent needs particular care, which timing detail cannot be shortened without risking failure. What might once have taken an individual researcher years to discover through personal trial and error could now be learned in a matter of weeks, absorbed from the accumulated experience of everyone who had faced the same small, tedious obstacles before them. As this chapter of standardization settles toward its close, imagine a shelf of neatly labeled reagent bottles, purchased rather than painstakingly homemade, each one a small monument to years of collective refinement that had transformed a fragile, unreliable laboratory curiosity into something closer to a dependable, everyday tool. The methods were no longer confined to the small number of laboratories that had first developed them. They were beginning quietly and steadily to spread. As these methods spread outward, something subtler than technique began to change as well. The very culture surrounding this kind of work. What had once been the province of a handful of specialized laboratories was slowly becoming a shared discipline, complete with its own conventions, its own shorthand, its own quiet etiquette for how new tools and new results ought to be shared with the wider community. Picture a gathering of researchers in a modest lecture hall, slides projected against a plain wall, presenters describing not dramatic breakthroughs, but small, careful refinements, a slightly improved method for introducing genetic material, a slightly more stable carrier vessel, a slightly more reliable way of confirming that a particular combination had been assembled correctly. These gatherings, unremarkable as any individual talk might have seemed, served an essential function, allowing standardized practices to be discussed openly, compared directly, and gradually converged upon by a community that had, only a decade earlier, been scattered and largely unaware of each other's parallel efforts. Consider the significance of shared reference collections beginning to take shape during this period. Centralized repositories where researchers could deposit carefully characterized tools and materials, making them available to anyone who requested them, rather than requiring each new laboratory to painstakingly recreate every reagent from scratch. Ordering a specific, well-characterized tool from such a repository, rather than attempting to isolate and purify it independently, saved enormous amounts of time and reduced the kind of subtle variability that had made early reproduction efforts so frustrating. A discipline that had once depended entirely on individual craftsmanship was slowly gaining something closer to shared infrastructure. Picture the careful, almost bureaucratic work of establishing common naming conventions, ensuring that a tool referred to by one name in one laboratory was recognized as identical to a tool referred to by a slightly different name elsewhere. This kind of standardization might sound dry, even tedious, compared to the excitement of earlier discoveries, yet it mattered enormously. Confusion over naming, over exact specifications, over which version of a tool a particular study had actually used, could waste months of a researcher's time chasing a discrepancy that turned out to be nothing more than inconsistent labeling. Establishing shared, precise conventions was quiet work, but it was foundational work. There is a particular satisfaction in imagining the accumulated weight of small courtesies extended during this period. One laboratory sending a carefully prepared sample to another with a handwritten note describing exactly how it had been stored and handled, ensuring the recipient would not have to rediscover, through frustrating trial and error, lessons the sender had already learned the hard way. This kind of generosity, repeated countless times across countless laboratories, is easy to overlook in any broad account of scientific progress, yet it represents the quiet connective tissue that allowed an entire field to mature far more quickly than any single laboratory could have managed alone. Imagine also the emergence of shared training programs, workshops where researchers from smaller or newer laboratories could travel to learn standardized techniques directly from those who had helped develop them, practicing careful pipetting technique, careful incubation timing, careful interpretation of the faint glowing bands that confirmed a successful combination. These workshops, often held in modest teaching laboratories rather than grand facilities, quietly trained an entire generation of researchers in a shared, consistent set of practices, ensuring that the same careful technique performed correctly in one country would be recognized as correct and perform the same way in another. As this chapter of standardization draws to its close, imagine a simple diagram pinned to a corkboard in a teaching laboratory, showing the now familiar steps of cutting, joining, introducing, and confirming, each step illustrated simply enough for a newcomer to follow along. What had once been a fragile, uncertain sequence of individually perfected steps had become, through years of shared effort, something closer to a common language, spoken with growing fluency by an entire, quietly expanding community. There are moments in any long story when the change taking place is enormous, yet almost nothing about the surface of daily life seems to shift at all. This was one of those moments. The tools for cutting, joining, and copying genetic material had matured. The protocols had been standardized and shared. And yet, for a long while, the work itself still looked much the same as it always had. Benches crowded with glassware, dishes stacked in incubators, notebooks filled with careful handwritten counts. The transition that mattered most was not visible in any single photograph. It was a transition in scale and ambition, happening quietly beneath a surface that still looked, to any outside observer, essentially unchanged. Picture a small machine, unassuming and boxy, sitting on a laboratory bench beside far larger and more established equipment. Inside this modest device, a sample was cycled rapidly through a sequence of precise temperatures, hot enough to separate two paired strands, then cooler, allowing short designed fragments to settle into place along each strand, then warmer again, allowing a natural copying process to extend those fragments into complete new strands. Repeated dozens of times in succession, this simple thermal cycle turned a single, nearly invisible trace of genetic material into millions of identical copies within the span of a single afternoon. A multiplication that would once have required weeks of careful cellular cultivation. Consider what this quiet acceleration actually meant in practice. A trace of material too small to work with directly, a sample barely worth mentioning under older methods, could now be amplified reliably into a quantity large enough to study, to sequence, to combine, to test. This was not a flashy transition. It simply meant that experiments once considered impractical, requiring impossibly large starting samples, suddenly became routine, opening doors that had been, for practical reasons, effectively closed. Picture also the slow, steady creep of automation into tasks that had once required painstaking manual repetition. Machines began handling the careful, repetitive pipetting that technicians had once performed by hand for hours at a stretch, transferring small, precise volumes of liquid with a consistency no human hand could quite match across hundreds of repetitions. The gentle whir and click of these early automated systems, moving methodically across a tray of small wells, represented a quiet handoff of labor, freeing careful human attention for the judgment calls that still required it. There is a particular kind of stillness that belongs to a laboratory running one of these automated cycles, the researchers stepping back from the bench they once occupied constantly, checking instead on a small display showing the current stage of a process, unfolding invisibly inside a sealed block of metal. The work had not become easier in any absolute sense. It had simply been redistributed. Careful human attention now spent designing the experiment and interpreting its results, rather than performing every single repetitive step by hand. As this quiet acceleration continued, the scope of what could reasonably be attempted began to widen. Techniques once reserved for the simplest organisms, easy to grow and easy to study, began extending outward toward organisms with far larger and more complicated sets of instructions, one careful adaptation at a time, each new organism presenting its own particular quirks that needed to be understood before existing methods could be applied successfully. Consider, too, the particular relief this quiet acceleration brought to researchers who had spent years rationing precious, hard-won samples, measuring out fractions of a fraction of a drop, terrified of exhausting material that might have taken months to collect in the first place. Suddenly, a sample once treated as irreplaceable could be copied and copied again, with a confidence that felt almost extravagant compared to the careful scarcity of earlier decades. Notebooks from this period show a subtle change in tone. Fewer anxious notes about conserving material, more notes about which of several possible experiments to attempt next. Now that supply was no longer the limiting concern it had always been before. As this chapter of quiet transition settles toward its close, imagine a small metal thermal cycler humming steadily through its final cycle late in the evening, its narrow display counting down the last few minutes of a program set hours earlier. No one is standing beside it now. The laboratory around it is dim and mostly empty, and yet, inside that unremarkable metal box, an enormous and consequential kind of multiplication is quietly, patiently taking place. As tools for amplification and automation matured, another kind of transition was unfolding alongside them, subtler still, concerned not with copying genetic material, but with reading it directly, letter by letter, in its full and complete sequence. This ambition had existed since the earliest days of understanding heredity, but only now, with the accumulated weight of decades of refinement, did it begin to feel genuinely achievable rather than merely theoretical. Picture a long, narrow gel suspended vertically between two glass plates, an electrical current pulling fragments of genetic material downward at speeds determined by their exact length, each fragment glowing faintly under careful illumination, arranged in bands so closely spaced that distinguishing one from the next required patient, careful eyes trained specifically for this task. Reading a sequence this way, one careful band at a time, was extraordinarily slow. A single afternoon of labor sufficient to determine only a small handful of letters in the vast, sprawling instruction set of any complex organism. And yet this painstaking method represented genuine progress, the first reliable way of translating the physical structure of genetic material into a readable sequence of symbols. Consider the sheer scale of ambition required to imagine applying this slow, careful method to an entire organism's complete set of instructions, a task that, at the pace of early methods, would have consumed more years than any single career could contain. This scale of ambition demanded not just patience, but genuine reinvention, faster chemistries, better detection methods, cleverer ways of automating the reading process itself, so that machines, rather than human eyes, could interpret the resulting patterns with speed and consistency no person could match across such enormous volumes of material. Picture a newer generation of instruments, sleek metal housings replacing exposed glass plates, fluorescent dyes replacing painstaking manual band reading, laser light sweeping continuously across a moving sample and translating the resulting flashes of color directly into a stream of recorded letters, hour after hour, without pause, without fatigue, without the careful squinting concentration that earlier methods had demanded. The sound of such a machine running through the night, a steady mechanical hum punctuated by the quiet click of an internal mechanism advancing the sample forward, became a familiar backdrop in laboratories pursuing this newly accelerated ambition. There is something quietly moving about the accumulation of sequence data during this period. Individual letters gathered one careful reading at a time, slowly assembling into longer and longer stretches, fragments overlapping and aligning until entire regions could finally be reconstructed with confidence. Researchers spent long hours at computer terminals during this stretch, screens filled with dense strings of letters, searching for patterns, for overlaps, for the subtle errors that needed correcting before one fragment could be reliably joined to the next in the final assembled sequence. Imagine the particular satisfaction of watching two independently sequenced fragments align perfectly at their overlapping edges, confirming that both readings, produced separately and compared afterward, agreed completely across dozens of consecutive letters. This kind of confirmation, unglamorous as it might sound described aloud, represented the quiet backbone of the entire sequencing effort. Careful redundancy, layered upon careful redundancy, until confidence in the final assembled sequence became overwhelming, built not from a single reading, but from many overlapping readings, all pointing toward the very same conclusion. There is a certain patience required to sit with a screen full of dense, undifferentiated letters, hour after hour, waiting for a pattern to reveal itself, the way a shape sometimes emerges from a field of static once the eye has adjusted. Researchers of this period spoke of a particular kind of fatigue unique to this work, not physical exhaustion, but a quieter, more focused strain. The mind straining to hold long sequences in memory long enough to compare them meaningfully against one another. As this chapter of quiet transition reaches its final hush, imagine a darkened room filled with the steady hum of sequencing instruments running through another overnight cycle, their small indicator lights blinking in slow, patient rhythm, translating the physical structure of life into strings of readable text, one careful measurement at a time. Nothing about the room looks dramatic, and yet within its steady mechanical hum, an entire library of instructions was being copied down, letter by letter, for the very first time. No effort of this scale could be carried by careful hands and modest machines alone. As ambition grew, so did the need for something larger and more deliberate. A genuine infrastructure capable of supporting work that had outgrown any single laboratory, any single institution, any single country's resources, acting in isolation. Picture the construction of dedicated facilities built specifically to house sequencing instruments by the dozen. Entire rooms lined with identical machines running in careful synchrony, their combined output requiring an entirely new kind of logistics simply to manage. Technicians moved between rows of humming equipment on scheduled rounds, replacing depleted reagents, loading fresh samples, monitoring displays for any sign of malfunction, choreographing a continuous, round-the-clock operation that bore little resemblance to the solitary, patient benchwork of earlier decades. The scale itself had become a kind of infrastructure, requiring careful planning simply to keep so many instruments running smoothly in parallel. Consider the quiet, unglamorous work of building the systems needed simply to store and organize the resulting flood of information. A single sequencing run could generate more raw data in a single day than earlier researchers might have generated across an entire career. And all of that data needed somewhere to live, some system capable of storing it reliably, retrieving it quickly, and protecting it from the kind of quiet corruption or loss that could unravel months of careful laboratory work in an instant. Rooms filled with humming storage equipment became as essential to this infrastructure as the laboratory benches themselves. Climate-controlled, carefully backed up, tended by specialists whose entire careers were devoted to keeping this growing mountain of information safe and accessible. Picture also the careful development of software capable of managing this scale. Programs designed specifically to align overlapping fragments, to search for meaningful patterns across enormous volumes of text, to flag likely errors for human review, rather than requiring every single letter to be checked individually by eye. This software did not emerge fully formed. It was built gradually, one careful improvement at a time, by researchers who were as often programmers as they were biologists, learning to translate biological questions into the language of careful, efficient computation. There is a particular kind of collaboration that belongs to this period. Biologists and computer scientists working side by side, each bringing a different vocabulary and a different set of instincts to the same shared problem. A biologist might understand precisely what pattern needed to be found, while a computer scientist understood precisely how to search for it efficiently across an amount of data too vast for any human eye to review directly. This blending of disciplines, awkward and unfamiliar at first, slowly matured into something genuinely new, a hybrid form of work that belonged fully to neither field alone. There is a particular hush that settles over such a facility during the quietest hours of the night shift. The ordinary bustle of daytime staff, replaced by a smaller crew moving carefully between rows of equipment, checking readouts by the soft glow of small indicator lights rather than overhead fluorescence. It is easy, standing in such a room, to forget the scale of what is actually taking place. So ordinary does the humming equipment appear. And yet, within that ordinary hum sits an effort larger than anything a single laboratory bench could ever have supported alone. Imagine the careful planning required simply to fund and coordinate infrastructure at this scale. Proposals drafted and reviewed, budgets negotiated across institutions with different priorities and different constraints, schedules coordinated so that facilities built in different locations could contribute their output toward a single, shared, carefully organized whole. This kind of coordination, dry and administrative as it might sound, was every bit as essential to the larger effort as the laboratory work itself, the quiet scaffolding that made ambitious, large-scale collaboration possible in the first place. As this chapter of infrastructure settles toward its close, imagine a long corridor lined with humming equipment, cables bundled neatly along the walls, small indicator lights blinking in steady, patient rhythm through the quiet hours of an overnight run. What had once fit comfortably on a single laboratory bench had grown into something closer to a small, dedicated industry, built specifically to support a task that no single set of hands, however skilled, could ever have completed alone. As this new infrastructure matured, it required a different kind of discipline as well. Not just technical, but organizational. A way of ensuring that work performed in one facility could be trusted, verified, and built upon by researchers working in an entirely different facility, often on the opposite side of the world. Picture the careful development of shared formats for recording and exchanging data, agreed upon structures ensuring that a sequence recorded in one laboratory could be opened, read, and understood correctly by software running in an entirely different institution. Without this kind of quiet standardization, the flood of information being generated across so many facilities would have remained a scattered collection of incompatible fragments. Technically impressive in isolation, but nearly useless when it came to combining results from different sources into something larger and more meaningful. Consider the significant effort involved in establishing quality control procedures robust enough to be trusted across an entire distributed network of facilities. A single questionable result, produced by a single malfunctioning instrument in a single overlooked laboratory, could quietly undermine confidence in an entire shared body of work if it went unnoticed and uncorrected. Careful procedures were developed for flagging unusual results, for cross-checking findings against independent replications, for maintaining detailed records of exactly which instrument, which reagent batch, and which technician had been involved in producing any given piece of data, ensuring that any later question about reliability could be traced back to its precise origin. Picture the quiet, patient work of technicians responsible for calibrating instruments against known reference samples, running the very same well-characterized material through a machine on a regular schedule, simply to confirm that its readings remained accurate and consistent over time. This kind of routine, repetitive verification rarely produced anything noteworthy on its own. Its entire value lay in its consistency, in the quiet reassurance that instruments behaving exactly as expected, day after day, could be trusted to produce reliable results when it mattered most. There is a particular kind of trust being built during this period, not the dramatic trust of a single verified breakthrough, but the slower, more durable trust that comes from thousands of small, careful verifications accumulating steadily over time. A researcher reading a sequence produced in a distant facility needed to trust not just the final result, but the entire careful chain of procedures, calibrations, and cross-checks that had produced it. A trust built patiently through shared standards rather than through any single dramatic demonstration. Picture also the quiet, unglamorous role played by careful documentation during this period. Binders and later databases recording the precise history of every instrument, every calibration, every batch of reagent used across an entire distributed network of facilities. A researcher questioning an unusual result could trace its lineage backward through this careful documentation, checking whether a particular instrument had drifted slightly out of calibration on the day in question, whether a particular reagent batch had later been found to behave inconsistently. This kind of careful record-keeping rarely felt urgent in the moment it was performed, yet it became indispensable precisely when questions eventually arose. Imagine also the careful negotiations required to establish shared expectations around how newly generated information ought to be shared with the broader research community. Agreements about timing, about attribution, about ensuring that data generated through large, collectively funded efforts remained accessible to researchers everywhere, rather than becoming the exclusive property of whichever facility happened to generate it first. These negotiations, often conducted through careful, extended correspondence between institutions and funding bodies, shaped the underlying culture of openness that would come to define much of the collaborative work still ahead. As this chapter of infrastructure and trust reaches its quiet close, imagine a reference sample sitting in a small labeled vial inside a climate-controlled storage unit, drawn out periodically to confirm that some distant instrument, humming steadily through another overnight run, remained precisely as accurate as it had been the day it was first calibrated. The infrastructure being built during this period was not only physical, it was also, in a very real sense, a careful architecture of trust. No infrastructure, however carefully built, could remain confined within a single set of borders. The work being undertaken had grown too large, too demanding, too dependent on shared knowledge for any single nation to claim it entirely as its own. What emerged instead was something closer to a genuine global effort, distributed across continents, coordinated through careful, patient diplomacy as much as through careful, patient science. Picture a conference room filled with representatives from research institutions scattered across many different countries, translators working quietly in the background, careful negotiations underway about how contributions from each participating facility would be coordinated, credited, and combined into a single shared body of work. These gatherings were rarely dramatic. They involved long hours of careful discussion about technical standards, about funding commitments, about the precise division of labor needed to prevent unnecessary duplication of effort across facilities working toward the very same distant goal. Consider the logistics required simply to move materials safely and reliably across such vast distances, carefully packaged samples shipped between continents under precise temperature control, documentation accompanying each shipment to satisfy the differing regulatory requirements of each country involved. A single sample drawn in one laboratory might travel thousands of miles before reaching the facility equipped to process it, carried through a careful chain of custody designed to ensure it arrived intact, uncontaminated, and exactly as it had left its point of origin. Picture also the quiet cultural exchange happening alongside this technical collaboration. Researchers traveling between distant institutions to learn techniques firsthand, carrying back not just new methods, but new habits of thought, new ways of approaching familiar problems, shaped by an entirely different scientific tradition. A technique refined in one part of the world, adapted slightly to suit local conditions and available equipment, might return home altered in small but meaningful ways, enriched by its journey through an unfamiliar laboratory culture. There is a particular kind of communication that belongs to this global effort, careful written updates circulated regularly among all participating institutions, ensuring that a discovery made in one location could inform ongoing work in another before months were wasted independently rediscovering the very same finding. In an era before instantaneous digital communication had fully matured, this kind of coordination required real discipline, regular summaries drafted and distributed, progress carefully tracked across a sprawling, distributed effort that no single person could ever fully observe from any one vantage point. There is something quietly humbling about imagining the sheer number of ordinary working days folded into this global effort. Technicians on one continent finishing an evening shift just as colleagues on another continent were beginning their morning. The work itself never fully pausing, passed carefully from one time zone to the next, like a lantern carried hand to hand through a long night. No single person ever witnessed the entire relay. Each participant saw only their own portion, trusting that the portions before and after their own were being carried with equal care. Imagine the particular satisfaction, arriving gradually rather than all at once, of watching fragments of information generated independently in different corners of the world align seamlessly when finally compared, confirming that careful, separate efforts had indeed been converging correctly toward the very same shared understanding all along. This kind of confirmation, arriving through careful international coordination rather than through any single facility's isolated effort, represented a genuinely new kind of scientific achievement, one that belonged to no single place, but to a patient, distributed community spanning the globe. As this chapter of global connection settles toward its close, imagine a world map pinned to the wall of a coordinating office, small markers scattered across many countries indicating each participating facility, threads of colored string connecting them in a web representing the careful flow of shared information. The work being done had become larger than any single nation. It had become, in its own quiet way, a shared inheritance belonging to the world. A global effort of this scale did not only move samples and data across borders, it also carried with it questions, translated imperfectly from one language and one cultural context into another, about what this growing body of knowledge actually meant, and what responsibilities came bundled together with the ability to read and increasingly to edit the instructions carried inside every living thing. Picture a small gathering, held in a plain meeting room, somewhere between two larger conferences, where researchers from different countries sat together, not to discuss technical protocols, but to discuss something harder to standardize: the differing cultural and ethical traditions each of them carried into this shared work. A concept that felt straightforward in one tradition might carry entirely different weight in another, shaped by different histories, different religious and philosophical backgrounds, different collective memories of how scientific power had been used and sometimes misused in the past. These conversations moved slowly, translated carefully, revisited repeatedly, because getting them wrong risked far more than a failed experiment. Consider the particular difficulty of establishing shared guidelines across such genuinely different starting points. A guideline drafted with one set of assumptions in mind might need substantial revision before it could be applied meaningfully somewhere with a different legal tradition, a different healthcare system, a different set of public expectations about the proper boundaries of scientific inquiry. Committees convened specifically to wrestle with these differences, meeting repeatedly over months and sometimes years, drafting language carefully enough that it could be genuinely useful across many different contexts, rather than reflecting only the assumptions of wherever the drafting committee happened to be sitting. Picture the quiet, unglamorous work of translation in the most literal sense. Documents rendered carefully from one language into another by translators who needed not just linguistic fluency, but a working understanding of the underlying science, ensuring that a nuance present in the original phrasing was not accidentally lost or altered in its translated form. A single ambiguous word, translated slightly differently across different versions of the same document, could create confusion that took months of careful correspondence to resolve. There is a particular kind of respect embedded in this slow, careful process of cross-cultural coordination, an acknowledgement that no single tradition held a complete monopoly on wisdom about how this new capability ought to be used. Researchers who might have disagreed sharply on certain points nonetheless continued meeting, continued drafting, continued revising, because the alternative, allowing this work to proceed entirely without shared reflection, felt far more troubling than the slow, sometimes frustrating labor of building genuine consensus across genuine difference. Picture the long hallway outside such a meeting room during a break in proceedings. Delegates from different countries standing in small clusters, coffee cooling in paper cups, conversations continuing informally in whatever shared language happened to be available. Sometimes a third language belonging to neither speaker's homeland, pieced together with patience and goodwill. These informal exchanges, easy to overlook in any official record of proceedings, often did as much to build genuine mutual understanding as the formal sessions themselves. Friendships and working relationships formed gradually across repeated gatherings, year after year, conference after conference. Imagine also the role played by patient public communication during this period. Researchers stepping outside their laboratories to explain, in plain and careful language, what this expanding body of knowledge actually made possible and what it did not. This kind of communication required a different skill entirely from laboratory work, translating precise technical detail into language accessible to someone without any scientific background at all, while resisting the temptation to either overstate or understate what had actually been achieved. As this chapter of global connection reaches its quiet close, imagine a stack of carefully translated documents sitting on a conference table in several languages at once, each version reviewed and cross-checked against the others, representing months of careful, patient labor devoted not to any single experiment, but to ensuring that an entire diverse global community could move forward together, informed by shared understanding rather than divided by careless miscommunication. Every new capability eventually invites a quieter, more searching kind of question, one that cannot be answered in a laboratory or resolved through careful calibration. As the ability to read and edit the instructions of life matured, so too did a deepening conversation about what this capability actually meant, not technically, but philosophically, for a species that had spent so long simply discovering the patterns nature had already written, and now found itself in a position to write new patterns of its own. Picture a quiet study, shelves lined with books on ethics and philosophy, standing beside journals filled with careful technical results. A researcher sitting at a desk late in the evening, turning over a question that no experiment could settle directly. What responsibility comes with the ability to alter something as fundamental as the instructions carried within a living cell? Where does careful, considered improvement end? And where does something more troubling begin? These were not new questions in any absolute sense. Humanity had wrestled with similar tensions around every major new capability throughout its long history, but they arrived here with a particular weight, given how directly this new capability touched the very definition of what a living thing was. Consider the careful distinctions that began to take shape during this period between addressing conditions that caused genuine suffering and pursuing changes driven by preference rather than need, between interventions that could not be passed to future generations and those that could, between work conducted with careful oversight and work that might proceed without any meaningful scrutiny at all. None of these distinctions arrived fully formed. Each was debated, refined, and debated again, in conference halls, in academic journals, in careful policy documents, drafted by committees representing many different perspectives and many different values. Picture the particular difficulty of these conversations when they move beyond small groups of specialists and into the wider public conversation, where careful nuance was sometimes lost amid simpler, more dramatic framings. Researchers found themselves needing to communicate not just what was technically possible, but what remained genuinely uncertain, resisting both unwarranted alarm and unwarranted reassurance, trying instead to offer something rarer and more valuable, an honest accounting of both the promise and the genuine unresolved questions still surrounding this expanding body of work. There is a particular humility that belongs to the most thoughtful voices in this ongoing conversation, an acknowledgement that certainty, in matters this consequential, was neither available nor entirely desirable. Better, many argued, to proceed carefully, revisiting assumptions regularly, remaining genuinely open to revising course as new evidence and new perspectives emerged, than to settle too quickly on a fixed position that might later prove mistaken in ways not yet visible. Picture a slow walk home after one of these long, difficult meetings, a researcher moving through quiet evening streets, turning the day's conversation over in their mind without any tidy resolution presenting itself. This kind of unresolved reflection was not a failure of the process. It was, in many ways, the entire point, an acknowledgement that some questions deserve to be carried thoughtfully rather than answered hastily, sat with rather than dismissed, revisited again and again as understanding continued to deepen with each passing year. Imagine also the quiet, personal reflection that many individual researchers carried privately, apart from any formal committee or public forum, turning over in solitary moments what it meant to have devoted a career to work this consequential. Some found this weight steadying, a reminder of why careful, patient rigor mattered so much in the first place. Others found it genuinely difficult to carry, a responsibility that followed them home at the end of even the most ordinary working day. As this chapter of quiet philosophy reaches its close, imagine a single lamp burning in an otherwise dark study, a researcher closing a journal article and sitting for a long moment in silence before finally switching off the light. Some questions, this chapter suggests, are not meant to be answered once and then set aside. They are meant to be carried carefully and continuously alongside the work itself. Philosophy, unlike a laboratory protocol, does not arrive at a fixed and final answer. It continues, unfolding slowly across generations of careful thought, each new development in the underlying science reopening questions that had seemed for a while reasonably well settled. As this capability matured, the philosophical conversation surrounding it matured alongside it, becoming less about whether such work should proceed at all, and more about the careful, detailed texture of how it ought to proceed responsibly. Picture a public forum held in a modest auditorium, researchers seated alongside ethicists, patient advocates, and interested members of the public, microphones passed carefully around the room so that every voice, regardless of technical background, could be heard and considered. These forums rarely produce tidy conclusions. They produce something more valuable in the long run, a shared record of genuine concerns, genuine hopes, and genuine disagreements, carefully documented so that future policy decisions could be made with a fuller understanding of the full range of perspectives actually held by the communities most directly affected. Consider the particular weight carried by voices belonging to those most directly affected by conditions this expanding capability might eventually help address. Individuals and families who understood far more intimately than any laboratory researcher exactly what was at stake in decisions about how carefully or how quickly this kind of work ought to proceed. Their perspectives, sometimes urging caution, sometimes urging greater speed, added a dimension to the conversation that technical expertise alone could never fully provide. Picture a parent standing at a microphone in one of these forums, describing not a technical concern, but a lived experience, the daily texture of caring for a family member affected by a condition that this research might, someday, help address. Testimony like this carried a different kind of weight than any laboratory presentation, grounding an otherwise abstract policy conversation in the concrete, specific reality of an actual life being lived. Picture also the slow, patient work of drafting oversight frameworks flexible enough to accommodate a rapidly changing field without becoming so rigid that they stifled careful, well-intentioned work, or so loose that they failed to catch genuine cause for concern. Committees responsible for this balance met repeatedly, revising language, incorporating new evidence, listening carefully to researchers explaining what had changed technically since the previous revision, and to ethicists explaining what new questions those technical changes raised in turn. There is something quietly reassuring about the durability of this ongoing conversation, its refusal to settle permanently on any single fixed position. Its willingness to keep asking difficult questions, even when doing so, slowed the pace of technical progress. This kind of patient, continuous reflection, easy to dismiss as merely administrative, actually represented one of the most important safeguards accompanying this entire expanding body of work, a standing reminder that capability alone was never sufficient justification for action. Picture the particular satisfaction, rare but genuinely meaningful, of watching a hard-won revision to an oversight framework finally adopted after years of careful negotiation, a single paragraph refined across dozens of drafts, each word chosen deliberately to balance competing concerns that had once seemed nearly impossible to reconcile. Documents like this rarely receive public attention, proportional to the effort invested in them, yet they quietly shape, in ways felt rather than announced, how an entire field conducts itself for years afterward. Imagine a careful balance being struck, meeting by meeting, document by document, between the genuine promise this capability held for addressing suffering that had persisted for generations, and the genuine caution required when working with something as foundational as the instructions of life itself. Neither impulse, considered alone, offered a complete guide. It was the ongoing tension between them, carefully maintained rather than resolved, that seemed to produce the most thoughtful path forward. As this chapter of continued reflection settles toward its close, imagine the quiet shuffle of chairs at the end of a long public forum, participants gathering their notes, exchanging a few final words in the hallway outside, carrying the evening's conversation home with them rather than leaving it behind in the room. The conversation was not finished. It was, by its very nature, never meant to be. Philosophy, unlike a laboratory protocol, does not arrive at a fixed and final answer. It continues, unfolding slowly across generations of careful thought, each new development in the underlying science reopening questions that had seemed for a while reasonably well settled. As this capability matured, the philosophical conversation surrounding it matured alongside it, becoming less about whether such work should proceed at all, and more about the careful, detailed texture of how it ought to proceed responsibly. Picture a public forum held in a modest auditorium, researchers seated alongside ethicists, patient advocates, and interested members of the public, microphones passed carefully around the room so that every voice, regardless of technical background, could be heard and considered. These forums rarely produce tidy conclusions. They produce something more valuable in the long run, a shared record of genuine concerns, genuine hopes, and genuine disagreements, carefully documented so that future policy decisions could be made with a fuller understanding of the full range of perspectives actually held by the communities most directly affected. Consider the particular weight carried by voices belonging to those most directly affected by conditions this expanding capability might eventually help address. Individuals and families who understood far more intimately than any laboratory researcher exactly what was at stake in decisions about how carefully or how quickly this kind of work ought to proceed. Their perspectives, sometimes urging caution, sometimes urging greater speed, added a dimension to the conversation that technical expertise alone could never fully provide. Picture a parent standing at a microphone in one of these forums, describing not a technical concern, but a lived experience. The daily texture of caring for a family member affected by a condition that this research might, someday, help address. Testimony like this carried a different kind of weight than any laboratory presentation, grounding an otherwise abstract policy conversation in the concrete, specific reality of an actual life being lived. Picture also the slow, patient work of drafting oversight frameworks flexible enough to accommodate a rapidly changing field without becoming so rigid that they stifled careful, well-intentioned work, or so loose that they failed to catch genuine cause for concern. Committees responsible for this balance met repeatedly, revising language, incorporating new evidence, listening carefully to researchers explaining what had changed technically since the previous revision, and to ethicists explaining what new questions those technical changes raised in turn. There is something quietly reassuring about the durability of this ongoing conversation. Its refusal to settle permanently on any single fixed position. Its willingness to keep asking difficult questions, even when doing so, slowed the pace of technical progress. This kind of patient, continuous reflection, easy to dismiss as merely administrative, actually represented one of the most important safeguards accompanying this entire expanding body of work, a standing reminder that capability alone was never sufficient justification for action. Picture the particular satisfaction, rare but genuinely meaningful, of watching a hard-won revision to an oversight framework finally adopted after years of careful negotiation, a single paragraph refined across dozens of drafts, each word chosen deliberately to balance competing concerns that had once seemed nearly impossible to reconcile. Documents like this rarely receive public attention, proportional to the effort invested in them, yet they quietly shape, in ways felt rather than announced, how an entire field conducts itself for years afterward. Imagine a careful balance being struck, meeting by meeting, document by document, between the genuine promise this capability held for addressing suffering that had persisted for generations, and the genuine caution required when working with something as foundational as the instructions of life itself. Neither impulse, considered alone, offered a complete guide. It was the ongoing tension between them, carefully maintained rather than resolved, that seemed to produce the most thoughtful path forward. As this chapter of continued reflection settles toward its close, imagine the quiet shuffle of chairs at the end of a long public forum, participants gathering their notes, exchanging a few final words in the hallway outside, carrying the evening's conversation home with them, rather than leaving it behind in the room. The conversation was not finished. It was, by its very nature, never meant to be. As this long story draws toward its close, it is worth pausing to consider the shape of the whole journey rather than any single chapter within it. What began with a farmer's careful touch along the husks of grain has arrived across countless generations of patient observation and careful refinement at a capability that would have seemed to that same farmer indistinguishable from something beyond ordinary understanding. And yet, the underlying spirit connecting every step of that journey has remained, in its essential character, remarkably consistent throughout. Picture, one final time, the quiet through line connecting a garden of pea plants to a laboratory bench crowded with sequencing equipment, connecting a stained slide viewed by lamplight to a global network of carefully calibrated instruments running through the night. At every stage, the essential method remained the same: careful observation, careful testing, careful willingness to be corrected by evidence rather than by assumption. Small steps accumulated patiently across enough time for something genuinely significant to emerge. Nothing about this long story unfolded through sudden dramatic leaps. It unfolded instead through the slow, steady accumulation of careful attention, repeated across countless individual choices made by countless individual people, most of whom never saw the full shape of the story they were quietly contributing to. Consider the particular kind of patience this entire journey required. Patience measured not in minutes or hours, but in decades and centuries. Patience that outlasted individual careers, individual laboratories, sometimes entire scientific generations. This patience did not announce itself. It simply persisted, quietly, through notebooks filled with careful counts, through faint photographic plates studied for weeks before yielding their secrets, through global coordination meetings that produced, meeting after meeting, only incremental progress toward the eventual understanding. Picture also the many hands this story has passed through: farmers and physicians, chemists and model builders, technicians running overnight sequencing cycles, translators rendering careful documents into many languages, ethicists and researchers sitting together in modest auditoriums, working through difficult questions without easy answers. No single figure carries this story alone. It belongs, in the truest sense, to an enormous, mostly anonymous collective of careful, patient people, spread across centuries and continents, each contributing some small, essential piece to a whole. None of them could see completely from where they stood. There is a particular kind of comfort in this way of understanding progress, not as a sequence of singular dramatic breakthroughs, but as the steady accumulation of ordinary care applied consistently, generation after generation, until something extraordinary gradually took shape. This same pattern, so visible across this particular story, echoes quietly through countless other long human endeavors, a reminder that the most consequential achievements rarely arrive suddenly. They accumulate slowly through the patient, unglamorous work of many careful hands. Picture, if it helps to close your eyes and imagine it, the entire span of this story compressed into a single quiet evening: the granary and the garden, The darkened room and the humming corridor, the conference table and the modest auditorium, all existing at once, layered gently atop one another like sheets of thin, translucent paper held up to a single soft light. Every careful hand belongs to the same unbroken thread of attention, stretched patiently across more time than any single lifetime could contain. Each contributor passing the work forward, gently, to whoever would carry it next. As the light continues to dim, and this long story settles finally into rest, imagine once more that quiet field at the edge of a river valley, the farmer walking the rows at dusk, running a thumb along the husks of grain. That same quiet attentiveness, carried forward and refined across countless generations, now hums softly through laboratories and sequencing facilities scattered across the entire world, still asking the same essential question that farmer once asked without any name for it at all. What is passed down, and how, and what, with patience and care, might yet be gently understood? The story continues, as it always has, quietly, patiently, one careful observation at a time. And here, for tonight, we let it rest. And so, the narrative of this assembly comes to its quiet conclusion. Whether you are still awake, or whether you have already drifted into a deep and peaceful sleep, know that you are exactly where you need to be. The world is vast, but tonight, its systems are simply the backdrop to your rest. Thank you for joining me here at Quietly Made. Rest now. Join us again tomorrow. Good night.